The current eDNA analysis pipeline is broken. It's a trade off. You can either use traditional tools like BLAST, which are reliable but painfully slow, taking weeks to process data, or you can use the new AI models, which are fast but often act as a black box, missing the nuanced biological context that scientists trust. This delay and uncertainty are causing us to lose on precious discoveries in a race against time, especially with biodiversity declining at such an unprecedented rate. Our solution? A hybrid intelligence system. We do not replace the old ways, we augment it. Sequence AI processes every single sequence of eDNA through a two parallel complementary pathway AI model and then fuses the results. We give scientists the speed of AI with the trusted, explainable results of traditional bioinformatics. It's the best of both worlds with no compromises. Here's how it works. As you can see, we have taken the sequences data of the 16 sequence ribosomal microbacteria, converted it into a sequenced DNA of the ATGC format, and run it using our AI pipeline. Firstly, we begin using the GPU model, then we start our pipeline. After pre-processing the data and training our DNA model, we run it through two epochs and then run it against the BLAST database. Here is where our AI model begins working. We run it simultaneously against different pipelines. On path A, we run the trusted BLAST analysis against the provided reference databases by NCBI. This gives us a high confidence interpretable matches. Simultaneously, on path B, we use our AI enhancement model where it begins the novelty engine. This doesn't just look for matches, it learns the language of the DNA. We use k-mers to analyze the GC content and motifs to generate a deep learning embedding that is a unique fingerprint for each sequence. For better user experience, these unique sequences that are resulted after a machine learning model runs are stored up to 5 total runs of every user session using the long short term memory used with the help of reinforcement learning. Now, we combine the confidence from the BLAST engine and the pattern recognition power of our AI model. A low BLAST score but high AI novelty signal is a massive red flag for potential discovery. Finally, our smart clustering takes all these potential sequences and groups them using advanced algorithms like HDB scan into candidate species clusters. We have built a powerful technical engine. As we can see here, after using the taxonomy with our AI enhancement, we used k-mers and then clustered them into novel reads. After the clustering is completed, we use artificial neural networks to find the biodiversity indices, Shannon, Simpson, evenness, and richness. After that, we generate AI insights, which creates a folder in our system output and then stores the insights into a JSON format. So we convert our AI outputs in the form of DNA sequences and parse it to the JSON format, which we then showcase in a user friendly manner on the dashboard using 3D interactive plots and graphs. As we can see below, this is a research friendly terminal output of how the technical nuances and biological terms are used to analyze the following data. In the data format of 16 ribosomal RNA sequences, we found a total read of 100 that was processed, out of which we found a cluster of one novel candidates. After the neural clustering, we found K-MERS having zero clusters, and neural networks identifying one possible treatment of novel creatures. Our average AI confidence score of novelty is zero point 0.5 out of 1. The Shannon index is in negative, which demonstrates that there is no one singular species or gene that is overpowering or dominating the field of the 100 reads that we processed. The species richness is 1, which is the highest. The ecosystem complexity is 0 because all the DNA taxes that we used were of 16 sequence ribosomes only. And the taxonomic discovery that could be potentially novel is 60%. This is our runtime. The AI mode was full and the output is saved into the JSON format. Now we run our user-friendly dashboard. Here, while this runs, I also want to bring to notice that to be able to cater to a larger audience, we have three AI modes: full, none, and light. Full is for deep length AI analysis. None is only for the users who are interested in exploring the field of eDNA, and light is only for the people who are just students or researchers interested in experiencing eDNA through the formats. This is how our dashboard looks after analyzing all the data we've read through our AI pipeline. We have classified it in such a 3D manner where you can easily understand the analysis. This is our main novelty distribution where you can see each of the 100 sequences that we processed using every data or set of it. You can see the potential depth at which our AI system thinks that it can be found, the novelty score, the confidence of how much the AI thinks that this is potentially true, and the GC content, which is something specific to the G and C nucleotides present in marine microorganisms found at this depth. This is our Shannon index, the value of our Simpson index, the species richness, and the evenness. These are the four parameters that are already used in real life to analyze the value of any nucleotide present in any kind of marine microorganism before studying them in depth. This is an overview of the total reads that we did. Below is our depth versus novelty. This graph tries to depict at what depth can you find the most novel creatures that are still untapped and unidentified by humans. So as we can see here, this suggests that at a depth of 5,000 meters, it currently has low novelty because this is an area that is highly untapped. What is available to humans and what can be mapped in the near future is something over here, which is 5,000 feet with extremely high novelty. And according to our AI system, 11 types of different species can potentially be found here. Then, out of the data of 16 ribosome microorganisms that we analyzed in the AI model, these are the type of organisms that we have found in it. Eukaryotic lineage, they may belong to abyssal protosta or any of the following other clusters which our AI thinks the nucleotides might belong to. And below is the sequencing of our AI's top 10 most novel possible creatures that we can be identifying through the following data set that we analyzed. Their novelty score, the depth at which they might predictably be found at, the confidence level of our AI, and how unique their GC content of the nucleotide is. These data are sequences IDs as following. Thank you so much. Team Estella.